So, good evening, dear participants. Welcome again to this 1 month comprehensive bioinformatics workshop on data science and machine learning with Python and genome-wide association studies. We have with us Priyanka ma'am. She has a vast experience in the field of bioinformatics. So, without wasting time, I am handing over session to ma'am. Ma'am, you are co-host for today's session now. Ma'am, am I audible? Ma'am, your voice is not audible. So I think there is some technical glitch, and let me find out. मैम की आवाज नहीं आ रही सर इस पे मीटिंग में। मैम इंट्रोडक्शन में दे चुका हूँ, आप स्टार्ट कर सकते हो मैम। ठीक, ओके। मैम योर वॉइस इज नॉट ऑडिबल।
Hello, can you hear me now?
Yes मैम, योर वॉइस इज नाउ परफेक्ट। यस मैम।
Yes, good evening, everyone. I'll be starting my lecture now. So I will first start with the basics and further we can whoever wants have any query can interrupt me or write in the chat so that I can know if you are stuck anywhere. Okay. So should I'm starting, okay?
Yeah, yes मैम, you can start now.
So you all can see my screen.
Yes मैम, your screen is visible.
Is my screen visible?
Yes मैम, your screen is visible.
Yeah, so today the topic which I'm going to cover is genotyping, phenotyping, and how the variants are identified. So before starting with this, first I would like to say that first you should understand what before genotyping we should understand how sequencing is performed, because genotyping experiment is also a type of sequencing experiment in which a genome is sequenced, but the chemistry is bit different from whole genome sequencing. So I would be discussing about genotyping first, about genome sequencing, right? So first, if you see that the basic for the basic, very basic students in the starting of their MSc, if you see that the base pairs, these ATGC in the DNA, they form the gene. This genes is integrated with the histone proteins, and it forms a nucleosome, and finally the chromosome is formed. This chromosome is present in the nucleus of the cell. So in the DNA, these ATGC pairs are present, which is sequenced during the sequencing run. Okay, and this is the very basic that what are these ATGC. So if you want to know, I'm discussing a little bit about how sequencing started. So the if we discuss about the main projects of sequencing, first human genome sequencing started in 1990, where Human Genome Project was launched, and then it was UK 10K Genome Project was there, in which the genome was to be sequenced for the humans. Then the Saudi Arabia 10K Genome Project came. Further, here both side you can see some UK 10K, 100K Genome Project was there, and then the Australia 100K this genome. So several genome project are coming because this field is very emerging, and it's very useful for the for the current research and current studies, all in all plants, animals, and everything, in all the types of organisms which are being studied. Okay, the latest, the India Indigen project, as you know, which genome project, which has been started by IGIB, National Institute of Genomics and Integrative Biology, IGIB, under the supervision Dr. Vinod Sakari and Dr. Sridhar Basu. This is the most recent project which is started in India. So the next thing I will be doing is telling you about what are the different generation of sequencing, what was the starting, what was being done, used for sequencing different platform in the first generation. The first generation was, I'm discussing few of the platform, not all, like Illumina High MiSeq was used, and sequencing in large gels or capillary tubing was there, which was limitation. Then came the second generation sequencing, in which sequence millions of clonically amplified molecules per run using an irreversible stepwise sequencing chemistry on an immobilized surface, which was used by life science technology, which is applied biosystem. Then there was Kaigen also introduced a gene reader. Then Roche, this sequencer, was initially very famous. This was a pyro sequencer during second generation sequencing, which is not at all used now. So with the time, the sequencer were also updated, were also modified, and the chemistry was modified, and such sequencer came which were more and more uh, cheap and generate higher amount of data. Then this is one of the third generation sequencing, what we call the next generation sequencing, also where the high throughput DNA sequencing technique, which reduce the sample size, cost, less time, massively parallel. This is a high-sec machine which it is being shown. It is of Illumina, where sequence which is highly popularly used, sequence of thousands of sequences at one time, and it produces enormous data. Now the next version of this is no is NovaSeq, which is currently being used for Illumina sequencing, which is for short read sequencer. For the long read sequencer, there is Sequel, which is it's a packed biosystem. Okay, So now I would like to compare different methods which are being used in the sequencing. The and I am comparing their why they are different from each other. Okay, this is a sequence different. They're different. These all are doing sequencing. These all chemistry are doing sequencing, but the method is different. Like single molecule real time sequencing is a SMRT, which we say, which is used by PacBio machine, which produces long reads. Ion semiconductor. See, if you see the read length in single molecule real-time sequencing, 2900 base pairs as the average size of a read. While in ion semiconductor based, it is of 200 base pairs. Similarly, if we compare it with the pyro sequencing of the Roche, which was old, it was having a size of 700 base pairs. Then the sequencing by synthesis, which is done by short read sequencer of Illumina, this chemistry of sequencing by synthesis, in which the size of the reads vary from 50 to 250 base pair. And then comes the sequencing by ligation, which is the chemistry of solid sequencing, in which this read size varies from 50 to 50 plus 50 base pair or 50 plus 35 base pair. Then the Sanger, which is the oldest method of sequencing, chain termination, sorry, which was and being used now also for some of the experimental validation, in which around 400 to 900 base pair read length was generated. So if we compare the accuracy of all these, you would see that around accuracy of this long read sequential SMRT, which is giving a very long read, the accuracy is not very high, but uh, uh, it is sometimes it can go to 87%, but sometime accuracy in accuracy mode, it also gives 99%. The iron semiconductor gives 98%. Pyro sequencing, which was earlier used, which was not cost effective, it used to give 99.9%. Illumina sequencing by synthesis was giving 98% accuracy, and the sequencing by ligation, that is solid, is giving 99% accuracy, while chain termination is also 99%. This is not used frequently because it is a very time consuming method, and it is you and it cannot be used for high throughput data generation. So if you see how many read runs are produced per run, that also varies. Like this has produced very high number of 5 million, and also in Illumina, which shows up to 3 billion of the reads being produced. Runtime, if you compare, then also you see that it takes 2 hours. Here it is discussing about the Illumina sequencer, which is HiSec, which is the old model. The newer model NovaSec is much faster in generating the reads. And then if you compare it with the other oldest technique, it's much more slow. And the cost, if you see, per 1 million base, how drastically it get reduced. Earlier it was, if you see earlier, Sanger sequencing was $2,400 for cost for 1 million bases. Now for Illumina it comes to so cheap, it's 0.05 to 0.15. It is highly cheap. So this is the reason why this different generation of sequencing platforms have come and why they are updating further and further. So the advantage is here it was that it is a long read, fast detects. This is less expensive, fast. This also has long reads. The Illumina sequencing by synthesis method give short read, but the quality is very high. It has potential for giving high sequence yield. Number of sequences will be very high, and if we compare it, solid has low cost per base, while the disadvantage is that for the SMRT is that it has low yield is not, number of reads are less, but the accuracy at the accuracy mode is high, as we discussed here. Then but the equipment can be the cost effectiveness is too not so good. It means the generation of reads is very costly using this instrument, that is SMRT. sequencing technology. Then if we compare sequencing, it has been almost absolute now, which was very expensive, and homopolymers error were there. And this Illumina is also a sequencing expensive experiment. No, every lab cannot take it. And this sequence solid is a slower method, and chain termination Sanger is impractical for larger sequencing projects. And the importance of genome sequencing, if you want to, because we have read so much about genome sequencing. Basically, genomics, in genomic study of the genes, we study in genomics, what we study is the genes, known different gene present in the genome, that we call the genes as genome. And in different, it includes the scientific study of healthy condition, like of related conditions, like it can be heart disease, asthma, any of the diseases are studies. And we also, it is also helpful to to conserve, protect, restore biodiversity, to create new benefits for the, to understand the biology and evolution. These are very important role of genome sequencing in all these aspects. So because I have explained you genome sequence, the whole genome means the machine, the platforms which are being used, the chemistry which is being used in during sequencing. It can be any type of sequencing. It can be that whole genome, or genotyping by sequencing, or it can be transcriptome sequencing, that is RNA sequencing. It can be small RNA sequencing. All these use when run on the, according to the platform it is being used, it runs on one of the chemistry which we have discussed. So now we will, because our lecture of the day is that, as I told you, that the lecture of my day is that I have to explain you genotyping. Genotyping is also a method of sequencing, that is, but genotyping in which we are not sequencing the complete genome. We are sequencing some part of it after fragment with a restriction enzyme. So sequence genotyping method is of three types. One is genotyping based on sequencing. Using sequencing, we can do the genotyping. The by array, we can do the genotyping. By PCR method also, a genotyping can be performed. Every method have its own advantage and disadvantages. All these three are being used. And this method, array based, is very popular in plants nowadays. And this is a method which is everywhere very popular, because sequencing give identifies every site, whether it is de novo or it is known. So it is a very it is such a good technique that it mines every unknown part of your genome. So what happens, what are the step in sequencing based genotyping? It is, first is sample preparation. During sample preparation, DNA extraction is done. Then after DNA extraction, its quantification takes place. Then its restriction digestion of that DNA takes place. Then the ligation of the, this, after restriction digestion, the ligation, that is just hold on. So after restriction digestion, for restriction digestion, restriction enzymes are used, that is PST, either PST1 or MSP1. This upon restriction digestion, like means cutting, that is the cutting of the whole, because the genome, whole genome of a cell is a chunk, is a bigger chunk. Now it is being cut by restriction enzyme, and then a different enzyme, are adapters are ligated at their ends. And then in this step, there comes the sample purification, sample cleanup, the PCR amplification, then final its quantification. This is a sample preparation step, what we call. Once the sample is ligated with the adapters and the sample is clean, PCR amplification is done, then its quantification is done. So in order to check whether it has that sufficient quantity of DNA which can be used for the sequencing based genotyping. Okay. So when the library preparation step comes during genotyping, that is, library preparation is basically preparing your sample so that they can run on the sequencing platform. So pooling of sample is done. Then the size selection is done. And after size selection means what is the range of base pair which you want to be used as your library for after pulling of samples. Then the quantification of that uh, uh, sorry, the quantification is performed using, this is mentioned picogreen. This is a method which is used in wet lab experiment. Then a quantification is how much is the concentration basically. Then adjust, then the concentration is adjusted and libraries are combined. Then the library is denatured, diluted, and a control is added in the library. And then sample sheet is sample sheet, then the details of the sample. Then this input is used on the loaded on sequencer, and the run begins on the sequencer once you add your sample sheet. And then the finally the FASTA files which are which contains the read after sequencing are generated. And then the further step of SNP calling, that is a variant identification, is done, which I will be telling you in further classes. Now what is the difference? The first I want to tell you that this sequencing based genotyping is of two types very popularly used. One is GBS, genotyping by sequencing, and one is RADSEq. Both methods are a bit different in the genotyping experiment. First I will discuss those two methods, and then we will come to the remaining two methods, which are exactly different technology. This is a microarray based technology. This was the sequencing, and this is a normal PCR which can be run on a normal real-time PCR machine. But this is sequencing based genotyping, which is highly popularly used, and it has two major methods which are being used. One is which is GBS, which is genotyping by sequencing. First I will discuss what is performed in that, how genotyping by sequencing is different from RADSEq. Okay, so here I feel my slide is completely visible. So in genotyping by sequencing, what is done that the the genomic DNA from different individual is first restriction digestion is done. Then the digested DNA from different samples, in that ligation of P1 adapter. I told you on the earlier slide that P1 is an adapter used during before running, before preparing the during the process of preparing the library of the sequencing. This adapter is added, and once the adapter is added, after that P1 adapter P1 ligated, this P1 is shown by blue color. This P1 ligated adapter fragments from different samples are now pooled. Pooling means combining, you can understand. Then it is sheared. Sheared means shearing is breaking the DNA into smaller chunks. And then size selection means if the DNA of your cell which is input, it is like, for example, it is of 1000 base pair, it gets sheared, then it becomes 150 base pair. Then we will select the size based on the requirement. Generally 300 base pair to 150 base pair of size is selected for the further library preparation step that is to be used for the sequencing run. Then the ligation of P2 adapter is done. The P2 adapter, the P1 was first ligated, then the shearing size, and then the P2. Then the further amplification is performed. And after that, only fragment which have P1 adapter at one end are amplified. Means those adapter which have P1 adapter at the end are amplified, and further the sequencing run data is generated. So now the second method I will be discussing, that is the RADSEq, which I said restriction. It is a restriction enzyme based method only like earlier, but here there is a this restriction enzyme which is a for the genome DNA. It is restriction digestion with a methylase sensitive enzyme. Earlier it was not so, it was not a specific methylase sensitive enzyme, but here it is a methylase sensitive enzyme in which the DNA is digested. After digestion of the DNA, the digested genomic DNA from different individuals, in which ligation of a bar coded of one bar coded and one common adapter is done. Here it is not P1 or P2 adapter. Here a common adapter is used, and one bar coded adapter which is different for different sample, that is used. Then these fragments are pooled and amplified on the Illumina flow cell. This is a very short, I'm explaining you the short method. This two methods are very important, mainly used for genotyping, uh, ge- for genotyping data generation. These two methods I will now, next slide, I will discuss and elaborate. This is a very short, crisp what is actually done during the library preparation for before running it on the sequencer. So if you understand by overview, this is the genotyping by sequencing method, which I've discussed first to you. So in this, what is there was there? These are restriction sites present. Okay, like in sample one, here is a restriction site. In sample, here is the restriction site. Then, and in sample two, this site is not there. Site is what? A region of ATGC, means where, which is, it depends on what your restriction enzyme is specific to. So if in sample one, there is a site here of restriction enzyme, it will cut here. But if in sample two, there is no such site, there will be a loss of cut. There will not be a cut here. Okay. So this is the actual what is being cut, that is being shown here in a very simple way. So now I will actually compare. This is a very, very good paper. If you get time and you actually want to understand the genotyping sequencing, how it is done, this is a paper in Nature, which says that whose title is Genome Wide Genetic Marker Discovery and Genotyping using NGS. In this, they have basically compared how genotyping the two main methods, that is, if you see here, it is mentioned the RADSEC and GBS, which are the very popular method of genotyping data generation in for sequencing run. So sequencing, so these two are compared, how these two are different. Earlier I have shown you individually, now I will show you comparatively, so that you understand why these two different methods are there, why what is what is different in these two. This also you should understand. And there is one more method, which is RRL, but it is not so popular, so I am not discussing about this method. So if you see what happens, that in RADSEC method, the there is first, you see that the once the adapters are ligated, as we saw that in this, in case of GBS, P1 and P2 adapters was ligated, while in RADSEC there is a common adapter, and one adapter is a barcoded adapter. Okay. So this after ligation of adapter, and there is difference in the adapter digestion ligation. Before that, the digestion step is common in all. Digestion is by a restriction enzyme. Here it was a methylase sensitive enzyme. Here it was a restriction enzyme, simple it was mentioned. And then adaptation ligators are ligate adapters are ligated, and then the pooling is mixing of the samples, because all samples are mixed at a specific concentration and then used for the library sequencing. Then after that, there is a step of random shearing. Random shearing is done generally to, so that the cutting the fragment of that fragment of DNA which has been cut by restriction enzyme are further cut into smaller pieces, but that is random. That is not specific size will be generated. It can cut anywhere. Then this step is actually not being performed here. Here the last step, finally directly PCR is done, polymerase chain reaction is put, but here random shearing is done, and the size selection is done at this step only. And again a third, like because as we saw in earlier slide, that P1 was added first, then after shearing P2 was added. So it happens here that later P2 is added, but here what happened, that both are added together. P1 and the sorry, not P1, the library, the specific adapter and the barcoded adapter.
Yes,
So
ma'am.
then comes the PCR chain reaction,
Yes,
in which the
ma'am.
amplification is performed. And if you see final how the library samples look like, you will see that if sample one, you should surely read this paper to understand an actual difference. Okay, so if you see this diagram below here, you will see that In RADSEC, how is the library? The library is containing these adapters and the stretches in between the samples. These stretches are also present in the library. Okay. While in GBS data, it is a very most cost-effective technique in which these fragments in between of these DNA adapter are very shorter compared to the longer length in the this RADSEC data. Okay, this because it is actually show comparing you the RADSEC with the GBS data set. See here only this part, this short fragments are there of 150 base pair, 350, but here the in RADSEC the fragment are longer, which is to be sequenced. So as I've explained you the two methods of sequencing for genotyping, now I will be explaining you the someone has asked me how restriction enzyme identifies sites. If do you know what the restriction enzyme does? Yeah, I will share the link of the paper. Actual restriction enzyme identifies a specific region in your like every restriction enzyme has a specific cut region, the specific sites. So if that means if it is cutting a ATGC stretch, or it is or some restriction enzyme is specific to some other stretch, depending on what region it identifies and cut, it will cut that part of your input DNA. Because they, like here it is two enzyme name mentioned, PSTI and MSPI. So both has a different cutting site. So whichever whatever is their cut site region which they recognize, because we know whenever you take a restriction enzyme, it is mentioned what region it identifies and where it cuts. So according to that, the cut is there in your sequence. Okay. So I'll proceed further. So now, as I told you that one was sequencing by genotyping method, other method was the other method was the array based method. Array based method, in this what happens, this is the array based method for genotyping experiment, in which the amplified DNA is loaded. It is like a microarray. Okay, the amplified DNA is loaded on a microarray type chip. This is a microarray type chip box in which final means initially first the DNA is prepared, and then it is loaded on the in this hybridization step. It will be actually loaded in the final array, but before that, in this sample box, it seems to be like an array, but it is a sample box in which different DNA samples are present, which are fragmented. In every you will see that in all the three types of method, DNA is first fragmented because the size is very big. So it cannot be. Someone has asked something. Okay. Okay, thank you. So then after the DNA is fragmented, why is the fragmentation step so important? Anyone can answer? Okay, I will tell you. Someone has answered. Short read sequencing. Actually, the fragmentation is there. This is not a sequencing method. This is an array based genotyping. I'm explaining you. There are three methods. One is sequencing, one is array based, one is PCR based, which is a normal PCR reaction is only performed. Array based genotyping is on an array, which is like a micro array. Here the fragmentation is performed because the chunk of the DNA which we are studying to identify to do the genotyping experiment and genotyping is performed to identify the variants, which we will be studying in our next class. So array based genotyping also, and other method also. First, this big DNA is broken into smaller pieces, that is fragmentation, so that it can be read by the uh, by identified by here there is identification by a probe which is present on an array. Here there is reading by the sequencer. So if the size of the DNA is very big, because we, as we know, the size of our DNA is bigger, then in that case we will not be able to identify the region or sequence it. That's why it is fragmented. Once it is fragmented, then a resuspension and a hybridization buffer is added to it, and a PCR reaction takes place. Once it is PCR takes place, its incubation is performed, and the DNA is precipitated. After that, it is hybridized to the array, that is the array based. There are many array for it, like it is there is a 90K rice array. There are different human arrays in which actually there, how does it get hybridized? If you see that there are, it is just like micro array, there are probes present on the floor of this array, and the input DNA binds to the complementary probes for which it's specifically designed. Every probe is designed for the specific region of a gene, which reflects the specific region of a gene. It binds and it shows that that genic region is present. And what is the sequence there? It is being read by this hybridization. Then after hybridization, ligation and washing is done. Means once it get hybridized, and then it's proper ligation, and then the washing, that is the extra molecules get washed, and then the data analysis is performed. And if we okay, if we perform the PCR based genotyping, then PCR based genotyping is a very simple method in which just like the normal PCR in our labs, DNA sample is mixed with the assay mix. Assay mix is basically the primer. Here the assay mix is for genotyping. For genotyping experiment, the primer is not a normal primer like we use in lab. It is a fluorescence labeled primer, and the primer, the is the two reverse and the forward primer are allele specific. Allele, why we are saying allele specific? Because here we are doing this PCR for the genotyping experiment in order to identify the allele. The allelic change, SNP, is present at the allelic position which gets changed. So we need to identify that that allele. So the primers are specific to the allele. And one is forward and one is reverse. Two primers are there and both are specific to the allele present. Oh, sorry. Then and they are labeled with a FRET reporting die. FRET reporting die, which is a FAM or hex. This is a method used by CASP, that is competitive assay specific poly, competitive allele specific PCR. So this method uses FAM and hex labeled dies. They give the prepared. We have to we have to send our sequence to LGC, which is the provider of this CASP enzyme. It adds the FAM and hex labels to your input DNA, and then they are used as an enzyme, and then the DNA polymerase is added, and ROX passive reference dye is also added. The MgCl2 buffer with TNTPs, which are used in a normal PCR reaction, are added. Then the thermal cycling, that is on PCR run, is done, and then it is detection is done based on fluorescent reading, because it is a fluorescence based reading, which is fluorescence means which is detected with the dye which is labeled FAM. So detection is done. Okay. And based on where it is present, clustering analysis assign those genotypes to the samples. Okay. So I feel someone has asked me something. Let me read. Thanks. Kindly find the PDF. Okay. What is the difference between genotyping using the method, these methods, and using the SNP array chip? See, okay. I explained you. I will explain you. See what is there, that this sequencing based method is reading you are taking your DNA, you are fragmenting it, you are ligating the adapters, and then you are sequencing every region of your input DNA. So you are actually identifying all the variants present in your DNA, whether it is known earlier or it is unknown. All the variants are identified. While in array-based genotyping, it is completely, if you compare a microarray which is used for a gene expression study, array has specific probes present on it. These specific probes which are present on the array, so whatever input you give, it will only detect those variants which are present on your probes, not those which are new or novel, which are new to your sample. So it is a very specific study, and but the accuracy of your call is very high in this, because you are very accurately identifying the variant in this method. While in this method, the identification of variant is comparatively less accurate, because here it is accurate, but compared to this, if it is 99.9, then this is not 99.9, because of sometime there is some sequencing error comes, so due to that. But here it is very specific binding, so accuracy is very high. But this is much more costly. The data generated is much lesser than this, and it only identifies the known SNPs, the known genic region which for which the probes are prepared. And this experiment is a very general experiment in which only a few SNP or variants can be identified, because for every variant you have to order this mix of enzymes, and this allele mix, which is a forward primer, allele specific forward reverse primers, these are costly. This is a patent of LGC, so you cannot yourself get it design. It is prepared by LGC, and their master mix and all cost is also high. And this is it is a very elaborate experiment, because if in data we get thousands of SNP, so we cannot put thousands of PCR reaction in a normal PCR machine. But we for that case we have to, for a high throughput seed companies, they have the machines um, which are provided by this LGC to run this genotyping experiment, in which it is a robotic system. I have not given the slide for it, but I can show you in the next class, in which you can run thousands of sample at a time in an hour. So that is being used by seed industries and many high throughput labs, but that is a uh, costly event, and it's a costly input for the lab. Every lab cannot afford it for so many reactions. So I will be now explaining you about the phenotyping. By phenotype,
Hello,
what do you understand? Anyone
ma'am.
can
Am I
tell
audible?
me?
Hello, hello. Yes, ma'am. Am I audible?
It is PST or MSTL. Okay, and it can, and either of these are used. And if you go in the depth of that paper, then you can read that whether any other is being used, because I am a computational biologist. I have not read in depth that how many restriction enzymes are used. So if you have interest, then you can go and read it. So if you because see, my topic today is genotyping, and we are understanding the genotyping, phenotype, and the variance. So because what if you understand very basic, like phenotype is what we see, the observed things, the observed biochemical, physiochemical, morphological characteristic of an individual. Like phenotype of someone is tall, someone is short, someone is normal, someone is a carrier for a gene. And the like if you see here, here for a very simple diagram which shows how phenotype, like here it is showing you, like this is the phenotype. Few plants are longer, few plants are shorter, and this is the phenotype what we observe by eyes. And what is the genotype? Sorry, what is the genotype? If you see, the genotype will be the the genomic sequence, that is the ATGC region, which we have identified by our genotyping experiment by sequencing or by array using that. So we will be able to identify what is the difference in these lines, why one is short, one is long. So if we see that in one of the set of lines, first three lines, there is presence of A at a position, while in other it is a presence of G. So this is a very basic example of an SNP, that how the what is SNP, how it is different, and what effect it is causing in the phenotype. This is a very basic example. The in-depth we will be studying in our further classes. So currently in plant and as well as animal sciences, phenotyping has become a very robust, and it's a very high. It is not just phenotyping 10 samples or 10 animals or 10 human beings. It is phenotyping at a very large scale. It is a high throughput. Just like if you have a genotyping experiment at a large throughput in your sequencing run, similarly the phenotyping also has to be on a large scale, because any in silico generated data, whichever, if we do not have a huge number statistically, identifying the correct result is difficult. Therefore every result has to find its accuracy, we do a high throughput experiments are preferred. So high throughput in this different modern technology, a 3D imaging is done for observing the the size of the disease, the different parameter which are observable, which is done with 3D imaging, the far infrared imaging, and a different type of imaging technique, fluorescence imaging, automated watering, spraying, hypersensitive image spectral imaging, and visible imaging. These are different methods of observing. Other than this also, there are many different methods for observing the phenotype. So I will proceed to the next part. If you all have understood what is genotyping, what is genotype, what is phenotyping, what is phenotype, then I will explain you a little bit about the variants.
Am I audible? Hello. Am I audible? Hello. Am I audible, ma'am?
SP sir, I think madam is not
Okay, okay.
listening, so kindly type your question in chat box.
I'm waiting for your answers.
So
So I got two answers. Yeah, okay. I'll be adding the disease associated case control study of the Jivas in my coming lecture. Okay. So first I will be explaining you, because it is the first introduction class. So my focus is to first make the students or the participants understand also case control for disease state. Yeah. Yeah, I understood your point. So you all can see my slide, I feel. So now the basically for about after genotyping and phenotyping, but no one answered me that what you understand. Okay, you can answer me anytime in my chat. So I will be explaining you about the variants. What actually are variants? Are the change in the nucleotide sequence at a particular position, which is a very general definition. But there are different types of variant. One is a single nucleotide variation, which is at a single point. Other, which is called the SNP in general. But and this is a single position difference there but while there are some larger changes which with there can be single change is SNP, but there can be also larger change like insertion, deletions, means there is a base which get added, other base which get deleted. There are inversion, means there's a reverse of the actual nucleotide. All these categories of variation belong to the structural variation. Okay, so our focus will be on SNP. In future we can have some training on the structural variations also, but here our focus will be on the SNP, single nucleotide change. So now if we how to classify, how to functionally identify that, how important they are. So there are some change in SNP which are silent or synonymous. That is that change. There is change in that a base, which change in a base in region, that is a mutation, is not having a effect on the amino acid. So these are called the silent mutations. And if there is a change in the change due to the mutation, there occurs a change in the DNA base pair, and that results in the change in the amino acid, then it is a nonsense or a missense mutation. Okay, then comes the uh, if the missense missense and non-synonymous mutation. Nonsense is that mutation in which the change in base leads to the incorporation of a stop codon, due to which it gets stopped. So due to this, there is a shorted protein, that protein gets truncated, then that leads to a nonsense mutation. Yeah, you are right. Thank you, Dr. Sandeep. So, and then the other type of mutation is the frame shift mutation. In the frame shift mutation, what happens that when a base get mutated at a position by the addition or deletion of the DNA bases, that affects the reading frame. See, if a base get added, like it was A, T, G, C, and between G and C a base get added, then the reading frame will change now, because reading frame is a frame consists of the group of 3 bases, each codon and each codon is for one amino acid. But if a addition or a deletion takes place, that leads to a frame shift change. So this is the called what we call the frame shift mutation, and the resulting protein are general many times non-functional, because sometimes might be the change the change of frame leads to another amino acid, but many times that doesn't able to code for amino acid, and that leads to a non-functional protein. And the frame shift mutation can be due to insertion, a type of insertion. Due to insertion it has happened, or deletion, or there is a duplication. Okay, so what if we explain the insertion, then insertion is basically increases the amount of DNA bases, in which insertion means an DNA is in a base is getting incorporated. Okay, increase, it's a very general definition is written, that amount of DNA bases in a gene as few bases are inserted. Increase the amount of DNA means number of bases in a sequence, in a gene sequence, get increased due to insertion of few bases. Then deletion, it can be few bases, can be one insertion, can be of one base, or it can be of more than one base also. The deletion, it is a removal of few bases. So the deletion can remove a stretch of DNA insertion, can remove a stretch of DNA insertion, DNA, that is the base pairs. And insertion or deletion can be small or large, as I told you. It can be a few bases, or one or few bases, or usually less than, but it is less than 50 base pair. It is not so big that it will completely truncate your gene. And then in any of these cases, the protein formed by the gene will not be functionally proper, because it has not the actual protein which we required, because there is a change in there will be a change in the codon. Okay, so and frame will also be shifted. So now the other type of observation is that duplication. If there is a duplication, means a region got duplicated of a DNA fragment, that needs to repeat it abnormally one or more times, means more than one fragment got duplicated. This type of mutation may change the function of the resulting protein. So this also leads, this mutation, a change, duplication of a DNA fragment also leads to the change in the function of the protein. So now what we have read, we can again revise it, that we read the normal. The silent normal means this is the normal state, the, the three proteins which was being encoded, proline, threonine. This was the normal state and the sequence was this. Once there was a change, the C got changed to A, due to which there was silent mutation. That silent because the protein encoded was same as the normal state. Then a mutation occurred at A changed to T. Then this protein got changed. Changed to serine. So this was a missense. That is a change in the protein sequence. Then if up here you see that before G, nucleotide got added. That is G got, that is the frame nucleotide got added and CAT is there. Now what happened that the frame got shifted. This is codes for histine. This codes for something else, not serine. So due to which this we call a frame shift mutation. Then comes the non-frame shift mutation, in which a complete three nucleotides got added, due to which the amino acid, a new amino acid got added. The earlier protein amino acid were same, uh, glutamic acid, proline and threonine, all three were there, and a new amino acid got added, that is alanine. So but the frame didn't shift, but the protein obviously will change as a new amino acid added. And sometimes there is a mutation which leads to a stop codon, like TAG. Earlier it was CAG, it got C converted into T, due to which there's a termination stop codon now. So this leads to the stop mutation. Now, as we also discussed about the insertion, deletion, duplication, we can see here again that in deletion leads to that these are the bases. Among these many bases, several bases got removed. Now it is just only 3 base pair left. Insertion, only 3 bases were present, many bases got inserted. Duplication, in which the 3 base pair got duplicated and they got doubled. Then the translocation step, in which the region of which was earlier the base pair were present here has got translocation, moved to the other position. So these are the different types of variants which we will be in the next class trying to study how these variants are identified in a sample data set. It is not, I'm not emphasizing that the sample data set is of X or Y. It is general. There is a reference genome. There is a GBS data which is of an organism is generated, and we will be identifying these variants, what variants we are getting. And two methods we will be using is either it is a reference based, that is for identification using a reference genome of these variants, and other method I will be telling is identification based on without a reference genome. That will be in the second class, then third class. And then here I have some alternative splicing, but I don't feel this will be so much required. Now I will tell you how a variant position, like what is how a nomenclature of the variant. Okay, so this is for human variation. Humans, there's a society, Human Genome Variation Society. They have standardized some parameters for how to define a variation. Okay, so that nomenclature we will be using. I will be describing you. See, to avoid any confusion, this Human Genome Variation Society has provides a nomenclature in which definition of multiple known basic variant type, the basic region, that is the basic variant types. That is what is this substitution is there, deletion, duplication, insertion, inversion, or conversion is there. Okay, once that all these basic information is there. For example, the standard format is HGVS format is that we give a reference ID, an ID to the change of that variation. What is their sequence? What is the sequence? It is A changed to T or T changed to what is being changed. Then the position. What is the location of that change? Okay. So here you see this is the reference ID. Then it says the what is the sequence type. By sequence type, sorry, it meant that either that is a coding or a non-coding. By that it means that, and by position change it is showing that at 630 position in your sequence G got changed to A. This is a very general standard HGVS nomenclature. So as I told you that here the sequence type is mentioned. So if it is a on a genomic chromosome sequence, then it is mentioned G here, as it is mentioned here that the different types of IDs given while describing a variant. HGVS considers a variety of reference supported reference sequence types are genomic is then it is G, mitochondrial then it is M. If it is DNA coding, then it is C, and if it is non-coding DNA, then it is N. So that varies. If you see here, if it is a genomic region, genomic variant, then here it is the reference ID, the G is there, then the location, that is the position, and G is changed to A. What is changed to what, that is mentioned. Then comes the, if you want that, this is also the genomic change, it is being shown. And here it says that it is it is in a transcript which is coding, so it is showing it is in the RNA, that is the nucleotide which is coding C, and here it shows that this change is identified at protein level, so it is P. And what arginine has been changed to the other protein, sorry, amino acid at this position. So these are the these are the different nomenclature which is being used for the different variants. I feel someone has asked something. Yes, I can share this paper with you. This is actually, it is a prepared document by the students only. It is a collaborative work that this paper, this document PDF was prepared, and I would also share few papers to understand Jivas, so that will benefit your course. It will benefit the course. So I hope for today I have given you a glimpse of what I wanted or you all to understand. So we can further discuss tomorrow in our next class, and I will be mailing you all the required as the document as discussed. Okay. So thank you everyone. Thank you for listening me patiently. No, in my next lecture, it will be all hands-on, because this lecture was not hands-on, because we in this first I had to explain you what we are going to do and why we are going to do. So that was the reason that I didn't do any hands-on here. Okay. Okay, thank you.
So thanks, ma'am. Now I am ending today's session, and I will share the video recording of this session within 1 hour to you. Thank you, ma'am.